Good morning everyone, I'm Listen. Today I'm going to present the topic Culture of Specialized Cells, Epithelial and Mesenchymal Cells. Before start the presentation, let me introduce my group members. She is Warda. She is Nadira. She is Dana. She is Ruhana. She is Salini. And she is Nafisa. Let's begin.
For our group, we will focus on mesenchymal cells. Before proceeding to mesenchymal cells, what are the differences between mesenchymal cells and epithelial cells? Mesenchymal cells are classed as stroma or supporting tissue, while epithelial cells are classed as parenchyma, the main functional cells within an organ. Mesenchymal is a special type of undifferentiated connective tissue, while epithelial line hollow organs and glands and make up the outer surface of body. Mesenchymal can differentiate into osteoblasts, adipocytes, myoblasts, or chondroblasts, unlike epithelial cells. Epithelial cells can produce mucus and some have cilia, which help remove foreign substances. Mesenchymal are fairly uniform small spindle-shaped cells and form a three-dimensional cellular network. Epithelial cells are polygonal-shaped, polarized in an apical basal orientation, and are organized into closely adherent sheets. Mesenchymal are vascular and can migrate easily, while epithelial cells are avascular and cannot migrate easily.
Now, let's move on to mesenchymal cells. Mesenchymal cells can be divided into two, structural cells and vascular cells. Structural cells consist of connective tissue, muscle and bone. Vascular cells consist of blood vessel, endothelium and smooth muscle. In this video, we will focus on connective tissue only. Connective tissue cells are survived most by mechanical and enzymatical explantation technique. May cultured in many simple media, particularly if serum is present. They are available commercially as fibroblasts. Although cells loosely called fibroblasts have been isolated from many different tissues and assumed to be connective tissue cells, the precise identity of cell is not always clear. Fibroblasts line produce type 1 and type 3 collagen and release it into the media. Although collagen production is not restricted to fibroblasts, synthesis of type 1 in relatively large amount is characteristic of connective tissue.
Now, how we want to prepare the connective tissue? Culture of fetal or adult fibroblasts can be prepared by several conventional procedures. They are primary explantation, warm trypsinization, cold trypsinization, and collagenase digestion.
Next, I will focus on the primary explantation and also collagenase digestion. Now, I will roughly explain about the primary explantation. The procedure for the primary explantation: first, we need to collect tissue sample in DBSS. Then we need to chop with cross scalpel to 0.5 to 1.0 mm. Then we need to wash by resuspension and settling 2 to 3 times. Then we need to transfer pieces to flask with pre-wetted pipette. The next step, we need to incubate overnight under thin film of medium. Then we need to make up medium to usual volume after 1 to 2 days. After about 1 week, cell can be seen migrating radially from the explant.
For my part, I will explain about the collagenase digestion. First, collect tissue sample in DBSS. Second, chop with fine cross couples to 2 to 3 mm. Third, wash by resuspension and settling 2 to 3 times. Then incubate finely chopped pieces in collagenous incomplete medium. Disperse by gentle pipetting, then transfer to tube and allow to settle. 7. For the first tube, collect supernatant cells and centrifuge. For the second tube, wash sediments by settling. For the first tube, resuspend cells in cross medium and seed flask. For the second tube, resuspend pieces in cross medium and seed flask.
Now, Dana will guide you the process of subculture.
So cells are usually passaged when its confluency reaches 80 to 90%. Initially, the culture media trypsin EDTA is pre-warmed to 37 degrees Celsius in water bath. Then, the cells are removed from the incubator and the media contained in the flask itself is aspirated off. Remember, this is the previous media. Then, the flask is washed with a new PBS and then is aspirated again. The word aspirated here means the action of sucking out fluid or gas. So in this case, it would be the action of sucking out the PBS itself. As you can see, after PBS is added, the flask is shook nicely so that the PBS touches all parts of the bottom of the flask. The PBS is then removed from the flask and discarded. Then, appropriate amount of trypsin EDTA is added to sufficiently cover the surface area of the flask. So that by means of sufficiency, it means that only a certain amount of trypsin EDTA is taken and it is added to the bottom of the flask and is ensured that the whole blot of the bottom of the flask is completely covered by trypsin. The cells are then rinsed in trypsin by gently swirling the flask around. Most of the trypsin are expirated off, except for a small amount that is just enough to form a thin layer over the flask surface. Then, the cells are incubated at 37 degrees Celsius or room temperature. The incubation time will actually vary depending on the cell type. The cells are then removed and observed under a microscope. When the cells appear sufficiently rounded, pat the flask to actually detach the cells completely. Then, appropriate volume of fresh medium is added, and it is then mixed well with the flask so that a whole lot of the cell and the medium are combined together properly. As you can see, the mixture is then sucked in and then released, sucked in and then released. This is to ensure proper mixing of the cells and also the new medium. The cells are next allocated to new flasks according to the recommended ratio of the specified cell line. This means that either it is being subcultured into two different flasks or a single flask is determined by the means of the project and also the specified cell line that is utilized in the project. So as you can see, before the cells mixture are completely sucked in, it was sucked and released to ensure mixture as well. The cells are then released into the new flask containing the new medium, which is the fresh media, of course. As you can see, each flask containing the newly subcultured cells are labeled accordingly before they are being incubated or before they are being put into the incubator. This ensures that there will be no mixture in the flask in case that we are sharing the same incubator, and this is also so that we can get to properly identify which flask actually belongs to us. Yes, and that is the end of subculturing adherent cells. Thank you.